Okay. Well, good morning to you all, and especially thanks to Professor Hendrikse for giving her availability on such short notice. My name is Matteo Rerizzo, and today I will present my research on the role of epigenetics in epilepsy, where I use the higher order multi-adaptive neural network model to attempt to understand the mechanisms to these epigenetic factors and investigate whether there would be a potential for therapeutic interventions. Epilepsy is an incredibly complex neurological disorder, and I feel like on the present day, we don't fully understand it, as traditional treatment often fail to address the underlying causes and mostly just treats the symptoms. Recent research suggests that epigenetic modification play a very important role in seizure development and their persistence. And in my thesis project, I apply Jan's computational approach to model these mechanics and explore how epigenetic therapies could restore normal function. So let's begin by outlining today's discussion. Today's presentation is divided into 6 main parts. First, I will provide a background on epilepsy and the role of epigenetics in this disorder. Following that, I will introduce the model and present the mathematical framework that underlies it. And after that, I will walk through my simulation results on both the base scenario and the therapy scenario. Lastly, we will discuss the key insight, future directions, and I will take your questions if any arises. So let's start with a brief introduction to epilepsy. Epilepsy is a chronic neurological disorder which is characterized by recurrent in addition to unprovoked seizures, and it affects people of all ages across the world. These seizures occur due to excessive and synchronized neuronal activity, which disrupts normal brain functioning. While anti-seizure medication are currently available, approximately 30% of cases develop pharmacoresistant epilepsy, which means that standard treatment just does not work for them at all. And for this reason, I feel like we need a deeper understanding of the underlying mechanisms and not just simply control the symptoms, which, as we see, does not really work. Recent research suggests that epilepsy is influenced by genetics, first off, environmental, but also by epigenetic factors. While genetics determines the predisposition to epilepsy and to seizure in general, epigenetic is more a bridge between the environment and the gene expression. And it is responsible for shaping the long-term functioning of neurons. So let's talk more about the epigenetic now. Epigenetics is a term that refers to changes in gene expression that do not alter the DNA sequence, contrary to other modifications such as mutations. These changes influence how genes are turned on and off and are directly responsible in the process like neuronal excitability and plasticity, which are incredibly important for epilepsy. The 3 main epigenetic mechanisms that are in play here are DNA methylation, first off, which is a chemical modification related to methyl groups. I will not go deep into the chemistry at play here, but I will say that methylation reduces gene expression by blocking transcription factors or by attracting repressive proteins. In epilepsy, DNA methylation can either enhance or suppress neuronal excitability, depending on the genes it affects. Next, we have histone modification. Histones themselves are proteins around which DNA is wrapped, and chemical changes to them alter the chromatin structure, which makes the DNA more or less accessible for further transcription. And lastly, we do have non-coding RNAs, such as microRNAs, and these regulate the gene expression after their transcription, but by binding to messenger RNA, so mRNA. And therefore they block translation, preventing the protein production later in the pipeline, or they promote degradation, destroying the messenger RNA before it can even be translated. These mechanisms together can contribute to seizure susceptibility by modifying neuronal excitability over time. But the way that this all work is a bit complex. So let's look at some molecular targets here. The microRNA play a crucial role in epileptogenesis, which is the process by which the brain becomes more susceptible to seizures over time. And they regulate the neuronal plasticity by targeting gene involvement in synaptic remodeling and excitability. For example, seizures can cause dysregulations of microRNAs that will lead to long-term shift in neuronal networks that will then lead to hyperexcitability that will cause more seizures and creating a feedback loop that leads to overall more seizures. Let's now focus on the two key genetic players in this big game, BDNF and LIMC1. BDNF, or brain-derived neurotrophic factor, is an important regulator of survival and plasticity of neurons. In simple words, it just makes synapses stronger, increasing the excitability of neurons. In the case of epilepsy, BDNF is often upregulated, which cause excessive neuronal firing and seizure persistence. LIMK1, though, called also limb kinase 1 in full name, regulates the dynamics of actin in dendritic spines, which are essential for the structure of the synapses. When LIMK1 behaves normally, it stabilizes these actin filaments and it maintains the proper dendritic spine structure, which in turn just supports the healthy synaptic function. But when LIMK1 activity is excessive, like in case of epilepsy, it leads to abnormal reorganization of these synapses and it just increases the excitatory connections. Other than the genes that are in play here, there's also the microRNAs that I cited earlier. And the two most important ones are 132 and 134. Why so important? Because they both work on dendritic growth or dendritic structure. 132 promotes their growth and the reorganization of the synapses, while 34 is found in the hippocampus and it affects their spine structure. There's been studies that show that the reduction of 134 levels has shown to suppress seizures, which is a very interesting fact to me. And it suggests that microRNAs in general could actually also be targeted therapeutically to modify seizure progression. Although epigenetic therapy is still an emerging field, it is now under extensive research. And in order to target epilepsy through epigenetic therapy, I have seen many approaches, but mainly two that have been explored. The first one is DNMT, which works on the methylation of DNA. It reduces the excessive methylation and restores the normal gene expression. And the second approach was histone deacetylase inhibitors, which increase the acetylation of the histones, which is one of the modifications that I explained earlier, making DNA more accessible for beneficial gene transcription. The goal of these approaches is to reverse faulty epigenetic mechanisms to a healthy state where they can perform in a positive way once again. And since epigenetic are indeed reversible, there is a potential to fix damaged systems in this way. Now that we've talked about the biological background, we can dive deeper into the computational model in use. The brain, particularly in epilepsy, operates across multiple timescales and levels of adaptation. It has fast dynamics ranging between milliseconds to seconds, up to long-term adaptations such as gene expressions or modification through epigenetics that last days or years at times. And given the multi-level complexity of epilepsy, where weights are fixed after training, uh, we cannot really capture the full dynamics with self-reinforcing processes. Instead, I decided to use a higher order adaptive model, which can modify its own parameters in response to changes in neuronal activity, which mimics a biological adaptation. It can also update the excitability threshold dynamically and adjust connectivity strength over time. The model has 6 key adaptation levels, from a very direct biological state to a high-level microRNA regulation. On the very bottom of this slide, we can see the pink level. That is the base level, which indicates the direct biological states. This is the foundation of the model, and it represents immediate physical and neural processes in the brain. From the very left, we can find con stress and con environment. And these processes are external stress and environment condition that might trigger epilepsy. During my simulation, I will then activate them in a period of time to see how it affects the whole model. Followed by them, just slightly right, there's SSS and SRSS, which are the sensory system that detects the stimuli and then makes the brain interpret it. After that, there's the node P seizure. That is the core seizure activity state. And when this one activates, the seizure occurs. This is the main focus on my paper and the simulations themselves. Followed by that, there is the activation of the amygdala and the hippocampus, the key brain areas involved in emotional processing, stress response, and memory. There's another 2 nodes down there, which are mostly to help represent the base for the transition from a healthy baseline state to chronic epilepsy, and to track the individual spikes where the seizure become persistent. Above the base level in blue, there's the first order adaptation that focuses on the excitability of neurons, where the brain adjusts the seizure threshold in response to repeated stimuli. And this means that over time, neurons become more sensitive, making it easier for seizures to occur. The second order, more in purple, introduces more biochemical regulations through the production of enzymes, which influence the neurotransmitters and further shape how neurons respond to excitatory and inhibitory signals. Third, in yellow, we have the adaptation through genetic levels, where the messenger RNA production begins to regulate the enzyme's availability. Lastly, not lastly, in fourth order, we do have the expression of the genetics, so BDNF and LIMK1 specifically, and their expression strengthens the pathways, contributing to a global hyperexcitability. Finally, on the 5th layer, we can find the epigenetic control, where we can find the context for the epigenetics in general, their mechanisms, and specifically the microRNA 132 and 134. Now that we've seen the model, let's look at the math behind it. So the model is based on a set of differential equations that describe how a state variable y evolves over time based on inputs and adaptive parameters. Let's break down the first equation. So on the left side of the equal, we can find the rate of change of y over time. We got a speed factor HY that controls how quickly Y adapts, followed by CY, a combination factor that puts together the inputs of other variables, X, through a function of the three that we can see on the right of the screen. W is the weight of the connection from X to Y, and YT is the current value of the state variable. The second equation, just below it, is a discrete time approximation of the first, where it updates YT in small steps of side delta t. Of the combination function that I used, we can find the logistic function, which is just an advanced version of logistic function. It takes multiple inputs, sums them up, and then applies a smooth transformation to make sure that the output stays between 0 and 1. It is controlled by a steepness parameter, sigma, which determines how quickly the output shifts from low to high, and a threshold, tau, which sets the point to which the change occurs. Well, essentially, it helps regulate when and how strong certain influences take effect. The step ones function is much simpler than this. It simply turns a value on and off for a specific period, which is what I did for the context for stress and environment in the base model. And the step mod function is similar, but it works in cycle. Instead of just switching once, it repeatedly turns on and off at regular intervals, which is what I use for chronic stress node to simulate the individual spike in epileptic seizures. So let's see at the simulation results. The first simulation showed how long-term stress can push the brain into a seizure-prone state. At first, everything is very stable, there's a baseline, but when stress is increased at time point 120, the brain becomes more excitable, leading to constant seizures over time. And normally the brain has a way to control this activity, but as we can see, this control system failed, and the system became hyperexcitable, and the seizure will persist even after the stressor is removed. Here, the stressor is removed and the seizure do not go back to the baseline state previous to the stressor. In the below image, there's a W states and the T states that also start incredibly high, and when the stressor starts, they plummet and never fully recover from that. In the second simulation, though, I introduced a new node, therapy, that affects the epigenetic mechanisms and specifically the microRNAs. And it is pretty much the same all the way to time 440, where the stress introduces this chronic epileptic seizures. But with the introduction of this therapy, there is a great gradual decrease in activation, and the system starts to return to normal. Some parts of the brain recovered quickly, while others took more time. But although the therapy didn't fully erase all the effects of the stress, the treatment could help bring the brain back to a stable state, as all the curves go back to an almost baseline state. So let's go over the key insights of the research. We've seen that chronic stress can induce lasting epigenetic changes that increase brain excitability and make it more prone to seizures over time. And these molecular adaptations help explain why some form of epilepsy persists even after the initial stressor is gone. Through the simulation, I observed that prolonged stress weakens the brain's ability to regulate the excitability, but reversing these epigenetic changes through targeted interventions can restore the brain to a more stable, less seizure-prone state. This suggests the promising approach of epilepsy treatment, but of course the model has its own limitation. Epigenetics, as we said, is a very complex interaction of genetic and molecular factors, and while I only focus on two genes and microRNAs, um, literature indicates that there's many factors that contribute to the epigenetic regulation of seizure activity as well. It was not feasible for MicroStrain to actually do a full study on all of these factors, but in future research, I would expand on this by incorporating additional genes and all the pathways to gain a more complete picture of on how the epigenetics contribute to epilepsy. Future studies should also use clinical data to tailor the model to individual patients rather than a more theoretical approach. And it should incorporate repeated intervention to see concrete long-term changes, to see it in a practical sense and not simply in a theoretical matter. With this slide, I reached the end of my presentation, so I want to thank you all for your attention, and I will be happy to take any of your questions.